This video deals with the applications of imaging mass spectrometry. Its contents include the definition of imaging mass spectrometry, the MALDI method, and the applications of imaging mass spectrometry, as well as a discussion. Imaging mass spectrometry allows for a detailed and direct characterization of the molecular structure of tissue components, whether animal, plant, or of other origins. Additionally, molecules can be qualitatively examined in their spatial resolution within tissues and in their molecular distribution, and relatively quantified. This can involve various groups of molecules, including proteins, complex carbohydrates, or lipids, which are simultaneously detected and particularly represented spatially. To understand imaging mass spectrometry, let's take the matrix-assisted laser desorption ionization MALDI system as an example. It consists of 3 units. First, an ion source generates an ion mixture from a substance sample, which then moves into a mass analyzer that separates the ions based on their mass-to-charge ratio. Finally, these ions reach the detector, which measures the ion current. The ionization of samples typically involves tissue samples, which are thin tissue sections containing analytes such as proteins, lipids, or complex carbohydrates. These analytes are present in a matrix, which is artificially applied. The matrix solution serves to ionize the analyte and later serves as the standard for detection. The laser vaporizes both the matrix and analyte, ionizing them as charged particles and carrying them away as vapor. This ion vapor is focused towards a mass analyzer, where an electric field separates the ions based on their charge. Different mass ions are then detected at different times. After detection, the ions are represented on a mass spectrum, with the x-axis showing the mass-to-charge ratio and the y-axis showing the relative quantities of individual ions. Matrix standards enable orientation and validation of our measurements. MALDI mass spectrometry imaging MSI is an advancement of the MALDI method to an imaging technique. The tissue section is initially rastered digitally, and each pixel in this raster is scanned sequentially. Thus, each pixel is individually ionized, representing the ion density distribution in the mass spectrum for each pixel. Each pixel on the resulting image corresponds to a point where the laser vaporized the sample, and behind each pixel lies a mass spectrum generated by the sensor. Today, pixel sizes range between 30 and 100 micrometers, allowing for the examination of small groups of cells and their molecular components and their comparison spatially and in terms of content. In MALDI MSI, not only frozen tissue sections, but also conventional paraffin sections can now be used. The only requirement is that the slides used are electrically conductive so that the tissue can be ionized later. The tissue sections are coated with the matrix solution, and it is important to achieve a homogeneous distribution on the section and to select the correct matrix and solvent for its dissolution. Typically, the dissolved matrix is sprayed simultaneously to ensure uniform ionization of the analytes. The selected matrix must be capable of absorbing the laser wavelengths and ionizing the analytes chemically with the energy absorbed. The solvent in which the matrix is dissolved must also be able to dissolve the analytes and must be used in small quantities to prevent delocalization or washing away of the analyte. The laser moves in a meandering pattern over the section, puncturing the tissue with its beam, so that both matrix and analytes from each quadrant are dissolved into an ion cloud and separated based on their mass and charge. The ions detected are visualized according to their mass-to-charge ratio and their localization. To understand a MALDI MSI device, let's look at the MALDI MSI device from Thermo Fisher at the Max Planck Institute in Bremen. It is used to study marine ecosystems, specifically the symbiosis of lugworms and microorganisms, by localizing metabolites in microorganisms. This promotes understanding of the degradation of plant material in the seabed. Similar projects include the symbiosis of plants and fungi. For us, the examination of myocardial chemistry may be more relevant. This involves a perfusion disorder or oxygen deficiency of coronary vessels. Here, localization of chemically typical molecules or normal typical healthy molecules in tissue helps us understand the processes that occur during myocardial infarction. It may also lead to the discovery of new markers for recognizing myocardial infarctions by examining the distribution of nourishing proteins or amino acids, as well as aromas or dyes in food. We can optimize the breeding selection and supply of breeding animals or crops, and thus provide better food. In tumor diagnostics, imaging techniques have long been used. However, the introduction of MALDI MSI allows for the direct correlation between tumor proteins and structural changes in tissue. By collecting and comparing tumor samples and physiological samples, new tumor-specific protein patterns, and possibly previously unknown tumor-specific structural changes in tissue can be discovered. Suspicious tissue samples are clearly examined for tumor RNA and gene expression analysis, potentially detecting post-translational changes in the mass spectrum. Possibly tumor samples can be distinguished from healthy ones purely mechanically by detecting certain peaks and distributions in the mass spectrum. Moreover, in elucidating the cell cycle, MALDI MSI can be a significant aid. Cells produce different proteins and other substances at different stages of their cell cycle, which we can analyze in the mass spectrum as peaks. Thus, we can locate cells in different stages in tissue and gain insights into the activity of their metabolism and the substances involved. This distinguishes not only living from dead cells, but possibly also particularly active, diseased from healthy cells. In addition to endogenous substances, drugs and their metabolites can also be localized and relatively quantified in tissues. The efficiency of drug uptake can vary greatly depending on the tissue type, and precise examination of drugs or their distribution in tissues is therefore important. Based on these analyses, drugs can be modified to target their site of action and be minimally absorbed by other tissues, ideally. Finally, I would like to compare the MALDI MSI method with two different established methods. The first is immunohistochemistry, where antigens, surface structures on cells, are made visible in tissues by specific antibodies and associated dyes. The number of possible analytes is limited by the number of antibodies used, and unknown analytes are not examined. Potentially in MALDI MSI, we can separate and represent all existing analytes, depending on the use of solvents and matrices. By comparing with databases, we enable the detection of unexpected analytes or entirely unknown ones, which are then captured and located for further examination, e.g., by extraction from the tissue. In the second step, I want to compare the MALDI MSI method with expression analysis. Gene expression analysis examines the expression of genes by quantifying RNA sequences in a sample. We know that RNA is the precursor of proteins and, so to speak, the intermediate step from DNA to the finished protein. In an exemplary representation, the volcano plot compares gene expression between two groups. We may be interested, for example, in comparing tumor cells and normal cells, as tumor cells and normal cells not only produce different proteins, but also different RNA sequences beforehand. The advantage of MALDI MSI here is the localization of the analytes in the tissue, which we do not have in gene expression analysis. At the same time, MALDI MSI can examine many molecular classes, not just the RNA that we examine in gene expression analysis. However, if we find suspicious proteins or distributions in MALDI MSI mass spectra, gene expression analysis is the next step. Because we not only want to know which proteins are present in what relative amounts, but also how they got there and what the possible genetic cause of their appearance in the tumor is. This is the interaction between the MALDI MSI method and gene expression analysis.